What's up, Mindsetters? It is that time. It's a Saturday with Looney and Yugan. We're doing life sciences. How are you doing, Yugan?
Good in yourself, Looney.
I'm good, thank you. So what are we doing today?
Well, today we're looking at life sciences, and it's grade 12s that we're focusing on.
Okay.
Lots of them are writing their prelims soon.
Yes.
So we're spending lots of time doing some revision. But
Okay.
we're focusing on paper 1 today.
All right.
So lots of questions from different types of papers. So I hope you guys are going to be tuning in and letting us know all the questions that you have as well.
Okay. Grade 12s, you heard it. Paper 1 prelim exam revision questions. Prelims are coming up, guys, so I hope you guys are excited and you're getting ready and you're preparing yourselves. Don't forget to hit us up on Twitter at LearnExtra, and our Facebook page is facebook.com/LearnExtra. We are posting all the notes and all the questions that Yugan will be going through on our Facebook page, so make sure you glued onto the page and you going through every question as Yugan is teaching it. So with all that said, Yugan, I think we should start with the lesson.
Thanks, Looney. So guys, we're spending this afternoon looking at revision of paper 1. Remember, it's that time of the year when lots of you are preparing for your prelims. The best way to do that is to take a section, revise a section, and then go and look at some past year papers. Look at the prelim papers, look at the examinations from the end of the year as well. And what we've done today, we've taken several questions, all relating to paper 1, but from different examinations. So we're looking at exams from the Eastern Cape, we're looking at some questions from KZN. So it gives you a broad perspective of what various possible questions are. So let's get straight into it. Right, so today we're looking at question 1, and this is an examination from the September 2012 GDE paper 1, and it's question 1.4. This normally comes in section A. Your 1.4 is a question that is there so that you attempt them and you score maximum marks in them. So very quickly, let's look at it. Colorblindness is a sex-linked trait caused by a harmless mutation. It's caused by a harmless mutation. I'm going to very quickly scroll up. The diagram below shows a family tree where the disorder is prevalent. So we're looking at a family tree, which is very similar to a pedigree diagram, which shows how the pattern of color blindness is inherited. And remember that we spoke about it being, we read here, it's sex-linked trait. Before we get into understanding what sex-linked trait is, we've got to remember that there are different types of patterns of inheritance. We have conditions that are sex-linked, and we have conditions that are autosomal linked, which simply means that if we talk about a sex-linked condition, it is found on the X and Y chromosomes, or your gonosomes. If we talk of a autosomal condition, it would be on all the other chromosomes except the X and Y chromosome. So what comes to mind when I look at sex-linked chromosomes or traits would be that I have to use the letters XX and XY, and that's fundamentally important. And lots of the learners make the mistake where they simply write down the letter describing the genotype without indicating or referencing that it's sex-linked. So we've got to be conscious about that. So let's look at the diagram. The diagram shows a family where the disorder is prevalent. I'll scroll down very quickly for you. Okay, so let's look at the key. Here we've got a set of parents, and on that we have an unaffected father. We have a carrier mother. Remember that a carrier mother refers to an individual that has both the alleles for it or is heterozygous. The word is heterozygous, that is scientifically correct. On this we have a key that shows us a square that is white or shaded, which is unaffected. We have a square that is unshaded, which is affected, and we have a square that represents partially shaded and half of it being unshaded, which represents the carrier. When we look at this diagram, it shows us the position or the locus of the gene on the X chromosome, and this individual is unaffected. We can clearly see a shaded or gray shaded area there. If we look at the female, she has a combination of affected and unaffected, which is basically now a carrier. So she's a carrier. And if we look at the offsprings, we've got 4 children, and each of them, they have their genotypes represented. That's a male, which has a unaffected genotype. Here we have a female who has been unaffected, and here we have a female who is partially affected and not affected at the same time, and we refer to those as a carrier. So she is a carrier. And here we have child 4, who is a boy who has been affected. So this individual is a male and is affected. So we've looked at the diagram, we've looked at whatever information they've given to us, and we've unpacked that information. Now guys, we've got to look at the questions. So we're going to refer to the questions very quickly, and let's see how we answer them. State one reason why color blindness is a sex-linked disease. Now we've got to give one reason why it is sex-linked. Now, as I mentioned in the introduction, we looked at the condition. It's called color blindness, and it is sex-linked. How do you know a condition is sex-linked? Well, when we look at the genotypes, or the key that is given here, we clearly see that the gene is found on the X chromosome. Here we're seeing it's found on the X chromosome. So we clearly can differentiate that it is found on the X chromosome. So the reason for it being called a sex-linked condition is that it is found on the X chromosome, and the X chromosome being an autosome, so it is being a sex chromosome, we call that a sex chromosome, and hence it is sex linked. Okay, let's look at the next question. We've got to explain why color blindness occurs more among males than in females. Guys, if we look at the occurrence or the positioning of the gene, so it's on the X chromosome, and if we were to write on genotypes of males and females, so a male would be that's the female. So females have, if you look at females very quickly, guys, females could either be that, they could be that, or they could be that. If we compare that to males, males have a single X chromosome, and remember that the gene is found on the X chromosome. So if we're using the letter G to represent color blind, that's normal color vision, and the letter small g to represent color blind or affected, then we could say that this male is normal and this male is, so that's a normal male, and that's a color blind male. Here we can look at, that's a normal female, that's a carrier female who is normal, so she is normal but she has the gene which she carries, and here we have an affected female. So if we look at the question, explain why color blindness occurs more commonly in males. The simple reason for that is, if we look at males, guys, they have a single X chromosome. Males have a single X chromosome. For the gene to be expressed, it is needed, we need only a single X chromosome. As opposed to in females, for a female to be colorblind, you need both the X's to be affected or to be to carry the gene, and hence we say that it is more current, or it's more frequent in males than in females, because males have a single X chromosome, and the possibility of that is higher in males. Let's look at the next question. Okay, if the symbol XG, and here they've given us a symbol, so we're supposed to use the symbol, represents the dominant gene for normal color vision, write down the genotypes of the father, mother, child 2 and child 4. So we've got to look at the genotypes of the father, the mother, child 2 and child 4. Let's very quickly go back. So being a father, guys, very quickly we know that he's male, so he has to have XY, and that's something that you must do first. The mother, being a female, has to be XX. Child 2, we got to look at the genotype. We're not too sure if they're males or females, so very quickly let's go back up to the diagram. So child 2 is a female, child 4 is a male. Child 2, if you look at child 2, she is a female who has, if you look at the key again, who is unaffected. So she definitely has a capital G. Child 4, who is a male, has a single X chromosome, and if you look at the key, guys, he's affected, so remember that he will be small g, x, y. And the parents, if we look at the parents are, so the father is an unaffected male, so he's going to be x, y, and we're going to put that down here. The female, who is XX, a carrier, would mean that she has the normal gene as well as the recessive gene. And hence we've got the genotypes of the father, the mother, and let's go down to child 2 and child 4, which is a male. Okay, we've done that. Let's move on to more questions. Okay, the question is, if child 4, let me extend the page, gets married to a normal woman who is homozygous, what is the probability of the daughters being carriers of the disease? Here we're coming across a word called probability. Probability simply means what is the chance? What is the probability that the offspring, in this case the daughters, will be carriers? The question is very specific about not the offspring, but specifically about the daughters. So we've got to look at what are the possibilities of the daughters being carriers. So very quickly, for a type of question like this, we've got to write down the genotypes of the parents. So in this case, child 4 is a male, and the male he's affected, is crossed with a normal woman who is homozygous. So a normal woman that is homozygous means that, firstly, we've got to say that she's a woman by putting down 2 X's. If she is homozygous and normal, it means that she has capital G, capital G. Cool. So we now have identified the genotypes of the parents. Now we've got to work out the probability of their daughters being carriers. And for this, we would need to use the Punnett square. So the Punnett square, guys, remember that we need to break down the gametes of this male. So we put that, we represent gametes using, so that's the parents. We write down the gametes of the parents. Remember to put your gametes in circles, so it just makes it easier for you not to confuse them. We then represent that, putting that into a Punnett square. Now often we get learners that prefer to do it mentally. For a question like this, there are going to be marks allocated for showing the methodology of how you came to your answer. So let's put down the genotypes. So let's put down, we're going to write down X small g, X, Y. That's for the male. X capital G for the female. And let's do this. So it's quite simple. Let's cross capital G first, small g, again, capital G, and then small g. If we look at the male, it's X first, he is capital G, and the Y, we bring that down, guys, capital G, and we transfer the Y there. Now we're looking at a completed Punnett square. Very quickly, I'm going to look at the question asked us to identify the females. So if we look at the females, it's these individuals here that we need to look at. If we look at the possibilities of females being affected, here we've got, let me write that down for you. We have these 2 females that are the offspring. Okay, and all we concern is in the females as the question. We're going to go up very quickly. The question says, what is the probability, guys, of the daughters being carriers of the disease? So what is the possibility or probability of the daughters? It doesn't say offspring. So we've got to look exclusively at the daughters only. So if we look at the daughters, what is the probability of the daughters being carriers? So let's read. 2 daughters. Is this daughter a carrier? Yes. Is this daughter a carrier? Yes. So of the 2 daughters, both are carriers. So what is the probability? The probability of that is 100%. So simply we would be able to say that there's a 100% probability that the daughters would be carriers of the condition. Very nice question to revise this section on. Now we're looking at another type of question, also based on genetics. This is obviously representing a pedigree diagram. This is adapted from September 2012. It's a GDE paper, question 1, it's obviously a question paper 1, question 2.2. Let's look at the question. We're going to read through again. We're going to highlight important words in this. Let's look at the word. Polydactyly, capital D, is a dominant autosomal condition or trait in humans controlled by a single, it should have been pair here guys, single pair of alleles. The pedigree shows a family in which the trait has occurred. Okay, so it's a pedigree diagram showing you how the history of this disease has been passed on, or this condition has been passed on. Answer the questions using capital D for polydactyl allele and small d for the normal allele. So basically the condition we're referring to here is polydactyly, and it's basically, poly means many, so it's many fingers, many more than what you normally have. So we naturally have 5 fingers, but often you see, or rare, you see an individual having an extra finger on both hands. And that's a condition known as polydactyly, where there is a presence of an extra finger. This is a dominant autosomal trait. So the condition autosomal refers to a gene that is found on the normal chromosomes except the X and Y chromosome. So it's an autosomal condition. And it is dominant, which simply means that if you have the condition, then you would have an extra finger. If you don't have the condition, we are normal. So I'm normal from this condition. I don't have the extra finger, so I would be representing my genotypes, representing using the letters small d. So let's very quickly look at the pedigree. Sure. Yep.
Can we take a quick break before
Sure.
we start with the question?
Is that time for break? Imagine.
Yes.
Time flies.
Mindset is we're going to take a very short break, but we will be back straight after this with all this fascinating stuff. I'm glued to the screen like you guys because I've never done this in my life. So I'm watching you guys teach this, guys. Oh, it's so interesting. So we'll be back straight after this break. Welcome back, Mindsetters. I hope you guys are tuned in and you're watching Life Sciences with Yugan and Looney. So we have a question here from Stefan Gonden, who's asking, why are sex-linked diseases commonly males?
So we did do that in the first question. The question is, again, I'll repeat that. Why are sex-linked diseases more common in males than in females? So if we look at colorblindness as an example of sex-linked disease, it's more common in males than in females because, as I said, for a male to be affected, a male has, I'm very quickly going to extend the page. I'm going to go back just to revise this section. I think it's important. It's an important question, guys. And if we deal with it now, if we look at this diagram here, if we look at this diagram here, we can see that for a male to represent the genotype, we use the letters X and Y. So what is the possibility of a male having the disease? If we take the letter G and small g representing colorblindness, for a male to be affected, he needs a single G, as you can see that. However, for a female to be affected, she needs 2 small g's. So what is the possibility of you tossing a coin and getting 1 head? It's 50%. But if you're having 2 coins and tossing both simultaneously to get 2 heads, what's the possibility of that? That's difficult. So you can liken the question to a similar example. So tossing a coin, you get a 50% chance of getting a heads or a tail. And that's why men are more likely. But if you take a female and you toss 2 coins and you get them both heads together, the possibility of that is 33%. So it's 1 in 3. And that is the reason why it's more current, more frequent in males than in females. So guys, remember that we're special as well. Am I right, Looney?
Yes.
Looney disagrees. Okay. So again, we're getting back to the pedigree diagram. And guys, remember that you will always get a diagram, you will always get a pedigree diagram, irrespective of the condition. It could be a sex-linked condition, it could be an autosomal dominant condition. The fundamental approach to this, the key approach to this would be to look at what the key tells us. So we've got to look at the key very quickly, we've got to unpack this. Look at the key and use the key to unpack what has been provided. So the key normally represents, so the squares represent males, the circles represent females. What we need to remember is that when we see a shade in it, the shade means that these individuals are affected. They're polydactyl. So I'm very quickly going to say, well, this individual is polydactyl, this individual is polydactyl. So these are affected individuals. The rest of them are normal. So based on the key, we very quickly, and you should do this. Take your highlighters, guys. Look at the key and very quickly highlight the affected individuals. I'm going to do that I'm going to go back to the key and let's look at what they've asked us. So they said polydactyly is a dominant autosomal condition controlled by a single pair of alleles. The pedigree shows us that. So it's a we've got to use capital D for polydactyl allele and small d for the normal allele. Okay, so it simply means that if you are normal, guys, if you are normal, which are these individuals here, so I'm going to change my highlighter. I'm going to put in some, let's say green. So all the individuals that are unaffected are these here. So I'm going to very quickly put down unaffected individuals, and that's the key approach. So I've identified, I've put down some color, and notice how color helps you to understand the pedigree diagram, to unpack the information. So simply by putting in color, I now can stand back, I can look at the question and say, well, these are the individuals that are affected, and the ones that have the green color are the individuals that are normal. But the key has given us information about the genotypes that need to be used. D would be affected, and small d would be normal. Okay, so remember that each of these individuals have a paternal chromosome and a maternal chromosome. So if we look at these individuals here, guys, so all the individuals that are normal will have to be small d, small d, small d, small d, small d. Okay, Looney is going, what's wrong with this oak? He's going small d all over. Okay, well, I simply am representing the maternal chromosome and the paternal. Remember that sperms and eggs come together, and that's how you get one from your dad, one from your mom. And that's how these individuals have got a small d and a small d, which have come from father and the mother. If we look at the individuals that are affected, for them to be affected, they must have one D. So I'm going to very quickly erase that. They must have one capital D. Okay, so we're going to very quickly give them a capital D to start with. The possibilities of individuals being affected, there are two possibilities. To be affected, you could have capital D, or you could have capital D, small d, which is basically homozygous or heterozygous. But we cannot at this stage conclusively say, and hence we just give them a capital D. And then we can work out what the possibilities of them either being homozygous or heterozygous, looking at the way the pattern has been inherited. It's very important for you to remember, and this is the key that I often tell the learners that I teach. When you're doing a pedigree diagram, look at the homozygous recessive individuals. Look at these individuals. Look at that. Let me just change my, let me use a nice pretty color. Let me see, let's look at this. If we look at this, now what does this tell you? When you look at small d, it simply means that one of the d's have to come from one of the parent, and this d comes from the other parent. So it simply means that if we go there, that both the parents are carriers or they are heterozygous for polydactyly, and hence we can identify that very quickly. So I'm not going to spend too much of time unpacking the entire pedigree diagram. Let's look at the questions and see what is necessary. But this is the approach. Look at the key, highlight the individuals that are normal, highlight the individuals that are affected, and very quickly look at the genotypes of the affected individuals and the unaffected individuals by representing them using the key or the genotype that has been given to us. Okay, so I'm going to go down to the questions and I'm going to see how we can answer them. What information can be deduced by the geneticist from a pedigree diagram? Now, who is a geneticist, guys? We got to understand and remember that a geneticist is somebody who works with pedigree diagrams, or even a genetic counselor will work with pedigree diagrams. The reason why they use pedigree diagrams, if you looked at that pedigree chart, you would see that it is to study the pattern of inheritance. So why would somebody want to know? It's very important. Suppose if we look at a condition like Down syndrome. Now, Down syndrome could be prevalent in a community. There could be a history of a parent or a grandparent or an uncle or niece having it. So when a couple decides to, when a couple marries and they decide on planning a family, they might need to know what is the possibility of us having an albino child or a child with a genetic condition. They would then need to approach a geneticist or a genetic counselor, who will then look at the history. He will look at the wife, he'll look at how she has inherited the condition, look at the parents, her grandparents, look at her siblings. They will look at the spouse, the male partner. They will look at his inheritance in terms of his parents, the grandparents, and see if the pattern. And then they will look at the probability of what the individuals could have in terms of if they were deciding to have children. And that's why it's done. It's done for planning, to be able to work out what the possibilities of having offsprings that are affected or that carry the condition. Okay, and I think it's becoming a more common practice. It's becoming part of good parenting, so that you understand, you know exactly how to prepare yourself and what are the possibilities of treatment if that condition does occur. So a geneticist basically uses this to determine the pattern of inheritance, to predict the possibilities of a condition appearing or being passed on to the next generation. We've got to very quickly write down the genotypes. Guys, the word genotypes, remember that there's two words that can be confused, the words genotypes and the word phenotypes. And I often remind my learners that phenotypes is the physical, P-H-Y, physical appearance, whereas your genotypes is your genetic appearance. So we've got to look at the genetic representation. So what are the genotypes of individuals A, B, and E? I'm going to quickly go up to the pedigree chart so that we can relate to, so it's A, B, and E. So we've got to look at A, B, and E. I've just put that down for a quick reference. Right, so let's identify individuals A, which is this here, B, and the third individual was individual E, which is here. If we look at A, how did we come to the deduction that A was capital D, small d? We said that firstly, because he's polydactyl, he's shaded, he has to have one capital D, which we have allocated to him. We're not too sure what the other D could be, but looking at the offspring, this is a normal male, and a normal male in this condition is represented using capital D, small d, small d. We asked ourselves a question, where does the small d come from? So one has to come from the father, and the other small d has to come from the mother. And that is how we conclusively can deduce that the parents have to be heterozygous for the condition, simply meaning that they are carriers. So individual A would be, go back to, individual A would have to be capital D, small d, which is the father. The mother also is capital D, small d, and we needed to look at individual E. And individual E, looking at the key, the unshaded individuals are normal. Remember that this is an autosomal dominant condition. So if you are normal, it means you are recessive, and for you to be recessive, it means that you must be homozygous recessive, and that would be the genotype of the individual that is male, who is shaded, who is unshaded. Okay, let's look at the next question. What percentage, guys, of the offspring in the fourth generation will be heterozygous? So let's go up to the fourth generation and we'll try and look at that question again. So read the question again. We've got to work out the percentage. In the fourth generation, that will be heterozygous. Remember, heterozygous simply means that for this condition, we've got to look at individuals that have this combination of alleles for their genetic representation. So we've got to look at what the possibility of, again, fourth generation being heterozygous. Go up to the diagram. Right. So we're looking at the fourth generation. Let's identify and zone out the fourth generation. That's the fourth generation according to the pedigree chart. So here we've got the first, the second, the third, and there the fourth generation. If we look at the fourth generation, we've got to identify what percentage of the fourth generation are heterozygous, which is this combination. So we've got to determine what is the percentage of them being here. Now, we've managed to do the genotypes of the individuals that are normal using the key that has been given to us. We now need to work out what are the genotypes of these individuals? We know that, looking at the key, that they are dominant for the condition, hence we've allocated to them a capital D. We now need to determine what the other allele is for each of them. How do we do that? Let's look at the parents. So these individuals, I'm going to clear up a bit here, guys, just so that we understand a bit, make it a bit less confusing. Okay, so right, so we're looking at, so this fourth generation is a result of a cross between the female here, who is, going back to my key, who is normal. For her to be normal, she is small d, small d. The male is affected, so he's definitely a capital D. We're not too sure whether he is homozygous or heterozygous, but we can determine that in a little while. Looking at these individuals, if you look at this female or even this male, the fact that they are normal means that they have inherited a small d from the father and a small d, from the mother as well as from the father, and hence the parents have to be of the following. So let's use this information now to work out what the genotypes of the male here and the female they are. We did confirm that they are dominant, but we need to allocate the other allele. And looking at the female parent, we know that she can only contribute, this has to come from the dad, although, oops, we got some, okay, so let's clear here, sorry. Okay, right, I'm going to try doing this again. Okay, so we said that, try this again, capital D, right. So let's look at this. So we said that they are, definitely, these individuals are, okay. So we got to ask ourselves a question. If the mother is homozygous recessive, it simply means that she can only contribute a D to each of the offspring. So the capital D comes from the dad, and the small d has to come from the mother, and then so what is the possibility, what is the percentage of the offspring being homozygous or heterozygous? And we would say there are 1, 2, 3, 4, and of the 4, 2 of them are heterozygous, and hence we would say the percentage of 2 over 4 is, that's half, which is 50%. So the answer that you needed to give was 50% of the individuals would be carriers. I think it's time for break. We're going to very quickly go for a break, and when we get back, we're going to tackle more questions on pedigree charts. So, Luni, to you.
All right, Mindsetters, we are going to take a short break. Just quickly, a shout out to Mazoba, Ane, Mercedes, and Stefan. Thank you guys so much. I see you have got questions. I'll ask you again during the break, and then we'll see you straight after this. Welcome back, Mindsetters, from that very short break. Matsobane, I see your question here, and he's asking you again. I observed a man can transfer his sex-linked genes to his grandchildren via his daughters. However, he cannot transfer it to his sons or their children. Is that true or false?
Sure, guys. Hope you understood the question. It's a very interesting question. The question is, he noticed that there's a characteristic that can be passed on from a man to his grandchildren, but not his daughters. Is it true or false? I'm going to very quickly represent this question now using colorblindness. Okay, let me just go up to the previous. Okay, so if we go all the way down here. Okay, I'm going to extend the page a bit. So if we have, if we have a male, suppose who is colorblind, and he marries a female who is normal, he might have a generation of offspring that are, the females will be carriers and the males will be normal. Okay, so you can have males that are normal, females that are carriers. In the next generation, you find that the males could be now affected, or they could be affected, they could be normal, and your females could be normal, or they could be carriers. Now, why does this happen? Remember that in the first, so that's the first generation, the second generation, the third generation. So the male is affected in the first generation. In the second generation, the males are normal. Why is that so? So firstly, if he is a male, and this is quite interesting, if he is a male, for him to be a male, what does he get from his parents? He has to get the Y from the father, which simply means that the father's affected or the recessive allele will not be passed to the males, which means that the second generation, in this case, the males will not be affected. So it has skipped the generation. Why? Because for this male to be, for him to be a male firstly, he has to have a Y from the father. And knowing that the way during gametogenesis, the sperms split up, you get either an X sperm with an X haploid chromosome, or you get a sperm with a Y haploid chromosome. And here, in this case, it's going to be that Y with the G, the X from the mother. And hence, the males in the second generation are normal. However, the females are carriers in the second generation. And it is because of the females that the third generation inherit the condition. Again, for the third generation sons to be males, they have to get the Y from the father. But to be affected, it is the carrier female in the second generation. And that's the reason why the males in the second generation do not show the condition. In the third generation it reappears. It's because the females in the second generation carried across to the third generation. And hence, the answer to that would be yes, it's true. Okay, Looney, is that the question does that answer the question for, is it Stefan?
Yes.
Okay.
No, it's Matsobane. Sorry, it's Matsobane.
Do we have another question?
One more question, yes.
Okay, cool.
I am not clear why individual A can have the genotype capital D, small d, and then he can only be small d, two small d's, because his partner is capital D, small d for any of their children to be normal. This is so because the X chromosome is masked in males. A little confused, and that's from Nikhil.
Nikhil? Is he referring to, I'm not too sure if he's referring to the previous question. Let's go back to
individual A, yeah, there.
So is he talking about the sex-linked condition? Which condition was he describing? Because if we're looking at individual A in this question, there is no A in this. It's just the sex-linked condition, and hence if we go back to the next question, which is polydactyly. Okay, I'm going to go down. I want you to read that. So it says.
Okay,
read that again for me.
I am not clear why individual A can have the genotype capital D, small d. And then he says he can only be small d, two small d's, because his partner is capital D, small d for any of their children to be normal. This is so because the X chromosome is masked in males. A little confused.
Okay. There's lots of there's conceptual understanding here.
Okay.
You shouldn't be looking at X and Y's because firstly this is an autosomal condition. Okay. So you don't have to Nikhil, if it's Nikhil, it's an autosomal condition. So we're not looking at X and Y's. It's simply representing the genotypes using capital D, small d in this case. Why are you I'm trying to understand why you're confused about A and B being heterozygous dominant. Remember that, as I said at the start of this question, it is an autosomal dominant condition, and all individuals that are shaded will be capital D. However, they could either be capital D that or capital D small d. The unaffected individuals will be, the normal individuals will be small d as I represented here. How can I conclusively deduce that that A and B are heterozygous? Would be by looking at their offspring. So again, if we look at A and B produce two offsprings, a male, which is a square, and a female. Remember that it is not sex linked, and hence we do not need to use X and Y. Being a male, he has to, and irrespective of the gender here, being unaffected, normal, he has to get this. Which simply means, if we use the recessive homozygous combination, okay, which is this, we got to ask ourselves, where does this D come from? The D has to come from the mother, and the D has to come from the father. And simply, just by looking at the D there and the D there, we can confirm. Okay, this initially was not there. I'm going to erase this. I'm going to erase that. So we first can do that, and we can say that. By looking at this, we can say, because the offspring, which is capital D, small d, is homozygous recessive, this D has to come from the father, and the other D has to come from the mother, and that is how we can conclusively deduce that A and B are heterozygous for the condition. Has Nikhil responded?
No.
Okay.
But
I
I'm
think
sure he
Nikhil
got
was a bit, I'm sure he's got it, but I think he was confused because he was trying to bring in sex-linked condition with this.
Okay.
So if it was sex-linked, it would be totally different. We would not be able to work out this pedigree using capital D, small d. We'd have to use X and Y's. Okay, so let's go further down. Let's see. Okay, I'm going to erase again. So let's see. Question D, based on the pedigree diagram, what is the probability of D having a polydactyl child if he married a normal female? Represent the genetic cross showing the possible phenotypes and genotypes of the F1 generation. And this, guys, normally counts for 8 marks. So this is a very complex question, but it's very easy if you understand the layout. So we've got to very quickly, we've got to find out the genotypes of D married a normal female. A normal female, guys. So remember that a normal female would be that. What is the problem of D? Let's look at what D was in the pedigree chart. So if we look at D, D is here. Before we start doing the pedigree cross, the pedigree diagram or the genetic cross, we've got to determine what D is. So D is, again, look at the key, guys. He is a polydactyl male, so he definitely is capital D. However, we've got to look at what the other allele is. And how do we do that, guys? Let's look at the offspring. Again, the offspring between the couple produced a normal male. Here as well, we're seeing a normal individual. Again, ask ourselves a question. For those small d's to come together, it simply means that one of the d's has to come from the father, the other d has to come from the mother. So both the parents are heterozygous. Now that we have the genotype of individual D, we can go ahead and do the genetic diagram. So I'm going to go down. I'm going to extend the page for you. We're going to go quite a bit. So we're working with, remember that, guys, there is a specific layout for representing a genetic cross. So a genetic cross is basically a system we got to use a system of codes and words to represent how we arrive at the F1 generation. So we've got to start with, I'm going to go down to the parents. In this case, the parents are individual D and a normal female. Okay, normal female. And this is individual D. Let's look at the genotypes of, and we call this the parent P1. So individual D, we said, is capital D, small d, and the normal female would be small d, small d. Now that we have the genotypes of the F1 generation, we now need to write down their gametes. So the gametes would be produced by these parents. We simply split them up, put them into circles, and we play the game called Punnett squares. So very quickly, we're drawing 3 rows, 3 columns. I enjoy this part, it's fun. 3 rows, 3 columns. Sometimes I get carried away, I want to do X and O's. It's very similar to that. And we go there, there. Let's do this cross. Capital D down, small d. Always remember to write the capital letters first and then the small letters. Here we have small d, small d, small d, small d. So here is the Punnett square of the F1 generation. However, the question does not end there. We've got to still write the F1. F1 means the filial generation. That's the F1 generation. And in this, we've got to write the F1 genotype ratio. So we could put down the ratio of genotypes. Here, if we look at this diagram, guys, here we can group these 2 individuals together. We can also group, let's see, let's use another color, these 2 individuals together. So we have 2, I'm going to change the color of my pen, 2 capital D, small d, 2 heterozygous, 2 homozygous recessive. we can simply write this down as capital D, small d in the simplest form, which simply means that for every, let's look at the condition, polydactyl individual, there is one normal individual. We still need to write the phenotype ratio. Okay, so the F1 phenotype ratio. Okay, so again, so we're going to write down this individual. If we go back to the key, this individual appears polydactyl, TYL, to normal. We've got to remember, guys, when you do a genetic cross or a genetic diagram, it simply means, we often use the word genetic diagram, the examiner will write the word, do a genetic diagram for the following cross. It simply means we've got to lay out a series of steps to show how we get to the F1 generation. I want to spend some time before we go into break showing you in terms of very quickly where the mark allocation for a problem like this would come up. So it's very important for you to understand where the mark allocation is, and I think that's important. So there would have been at least 2 marks given here to work out the genotype of individual D, because we had to go back to the pedigree chart. So there's marks for the parents, writing down the genotypes. There would be a mark allocation again for writing down the gametes of their parents. So that's 3 marks at least. If we come down a bit, then for showing the Punnett square and for correctly doing the crosses, there could be up to 3 marks for this. So putting down the parents, the gametes into the different columns, and then we go down to working out the F1 generation, the genotype ratio, which is given in its simplest form, and the F1 phenotype ratio, which is given in its simplest form, describing how they physically look. So here we've looked at a pedigree diagram. The pedigree diagram, we've looked at sort of a genetic cross. We've laid out a genetic cross. We've understood that you've got to lay out a genetic cross using parent, gamete genotypes, the gametes, showing the Punnett square. You then get the F1 generation, which you write down as the genotype ratio and the phenotype ratio. Okay. Examiners may ask you, what is the percentage of the offspring being polydactyl? In this case, there would be a 50% possibility of being normal and being polydactyl. So various questions can come up with pedigree diagrams. Prepare yourself, anticipate getting a question like this. You might get half of the question, but yeah, it's very easy to score marks in this section. So guys, we're going to go for a little break. I'm going to hand you over to Looney.
Alright, mindsetters, we are going to take a very short break. Nikhil said he understands now, so thank you for that. So don't go anywhere, guys. We will be back straight after this. Welcome back, mindsetters. I hope you guys are still enjoying life sciences. As I can see on the page, you guys are enjoying it. You're having fun. You're saying you can. It's the best teacher ever. So I'll take it straight back to him.
Thanks for the compliment. Right. So I know that definitely the guys are interacting, and that's important. Let us hear those questions. It's the best time to clear it out. So we're moving on to the next section. And remember, guys, in grade 12, you started off the year looking at protein synthesis, DNA structure of DNA, RNA protein synthesis. Remember, when you see a question, try and relate that to the context of a chapter. Remember that it's very good when you start identifying a question and putting that into a topic. It's easy for you to be able to remember content and relate that to the section that you've studied. So here we're looking at a question that's adapted from a September 2012 GDE paper 1, again in question 2. So let's look at the question. Study the diagram below on a process that occurs within a living cell and answer the question. So we're looking at a process here. We've got a diagram. Let's study the diagram and answer the questions. When we look at this diagram, guys, it looks like a golf ball. Am I right, Looney?
It looks like a
golf ball.
Yes.
But it's not. It's a nucleus. So that's the nucleus, guys. And obviously, if you've done the nucleus, the structure of the nucleus in grade 10, you would remember that those are the nuclear pores. Those are the pores on the nucleus. And here we're seeing a strand coming out, and here we're seeing something happening here. I'm not going to give too much of information about that, but we see a process happening here, and we see a little strand here. It's probably the strand that comes through here. So Let me be nice today and let's go through this. So here we're seeing the nucleus. Here we're seeing a structure that comes out. And out of the nucleus, guys, remember, the structure that comes out, single-stranded, and here's a clue, AUG, UUCA. Where do we find these nucleotides? Yes, we find them in mRNA. We're not going to find them in DNA, because in DNA, we don't have uracil. We have thymine. Okay, so here we clearly seeing that that's an mRNA molecule that comes out of the nucleus via the nuclear pore, and then it goes down into this. I'm going to try and add some color here. Around this structure here, I'm highlighting this little structure here, which you guys normally tend to draw like this. Okay, I call it the snowman, but it's a ribosome. It's got two subunits. You've got a big subunit and a small subunit. And what is the function of the ribosome? If you remember from grade 10 and grade 12, it is the protein synthesizing factory. So it is the organelle. Remember that it's a cell, so it's a little organelle that is responsible for the production of proteins. Very quickly to recap on protein synthesis, remember that protein synthesis simply means taking amino acids and putting them together into a chain. Here we're seeing the amino acids here, and they call asparagine and methionine. Quite complicated names, but you don't really need to remember them. You will be given them, you'll probably be given a table of them if you need to identify the amino acids. We're also seeing these quite complex looking structures here. Looney thinks it looks like a hair grip, but I can guarantee you it's not. It's ultra small. And that structure there, guys, is the tRNA. And if to quickly recap, the tRNA again is the transfer RNA, and to it is attached these nice looking amino acids. So the amino acids basically are transported by the tRNA to the site of protein synthesis, which is the ribosomes. So the ribosome is a site of protein synthesis. So we very quickly, here we also seeing a little label line, that's E. And I know all you bright grade 12s outside have already guessed what that is. And that is the anticodon. Awesome. If we go down, we see another label here, F. Okay, I'm going to change my pen because there's a conflict of color there. And that F basically is showing you 1, 2, 3. 3 adjacent bases on the mRNA molecule are referred to as the codon. And normally the codon and the anticodon are complementary to each other. G again is the structure here that came across, and so G is the mRNA molecule. C, we've identified C as the ribosome. So let's look at the questions. I've very quickly spent a few minutes unpacking the information here, writing down as much information that I possibly can to answer the questions. And that's what you must try and do. Very quickly, put in some labels, identify structures, parts, and then tackle the question. Because what happens is that it makes it easier for us to start answering the questions now. So let's look at the first question. Identify the process drawn above. If you look at the diagram, guys, the process drawn above, we can see, I'm going to quickly go back, we're going to see the mRNA molecule coming out, we can see it going to the ribosome here, and we can see that there are amino acids being put together. What do we refer to that process as collectively? The process of amino acid production coming together is called protein synthesis. And to remember protein synthesis, there are two parts to protein synthesis. The step that happens inside the nucleus and that happens outside in the cytoplasm at the site of the ribosome. So protein synthesis is the process that is happening here. It's made up of two processes. Transcription, which is a script, which is the message that is written. So it's written by the brain of the cell, which is the nucleus. It writes out a script, and that script is called the messenger RNA. Remember, the message is the script, and that messenger RNA carries the information to put the amino acids together. But, you know, the ribosome cannot read the mRNA message, so it will need to change the language. It translates the messenger RNA into amino acid sequence. So we call this process inside the nucleus transcription, which produces the mRNA, and we call the process here where the amino acids are put together and translation. So we're seeing protein synthesis as the process, but obviously we had to talk about the other processes involved in protein synthesis. Labels for A, B, and E. So very quickly, I've done that already for you. If we look at A, guys, let's find A. So it's slightly up on the diagram. We said that A was not the golf ball, as Looney was going to guess. It's the nucleus. B, guys, let's see B. B is here. Let me very quickly erase that. And B, we said, was the mRNA molecule, and the other part that we needed to identify was structure E, and we said that E was the anticodon, and that is found on the tRNA molecule. And to remember that it is complementary to the codon on the mRNA. Name the process responsible for the formation of B. So what is B? If we go back, B is mRNA. What is that process responsible for the formation of B? I just mentioned that. So obviously this is the messenger RNA. It is a script that comes out from the nucleus. So remember the word script, and hence the process that is responsible for B is transcription. So transcription produces mRNA. Awesome. So you guys are following me. I hope you guys are sending me questions if you don't follow. Okay, so we're going to go still with the same question. We're going to look at a few other questions on this. Let's see, this is a nice question. I actually like this question. Predict, we got to make a prediction about the outcome if the enzymes controlling the process at F were destroyed. Let's identify and recollect what are the enzymes. So remember that in replication we looked at a polymerase, but be specific about that. It's RNA polymerase. So RNA polymerase is responsible for replication. However, we're dealing with protein synthesis. So we have a polymerase and we call that, it's DNA polymerase responsible for replication. Correct that. When we look at protein synthesis, the enzyme that we're relating to is RNA polymerase. Okay, well there are three types of polymerases that we need to know, but we just need to remember that it's an RNA polymerase. Let's see. So predict the outcome if the enzyme, RNA polymerase, controlling the process was destroyed. Sure. And we know that enzymes are destroyed by change in pH and change in temperature. What happens if the enzyme controlling the process at F was destroyed? So what is F? What is happening at F? The codon and the anticodon are complementary, coming together to release the amino acid methionine. What will happen if this did not happen? This means that there was no methionine. We cancel this out. What would happen? Yes. If we remove an amino acid from a sequence of amino acid chains, what would happen? We delete one out. That's not going to form a chain of amino acids. If we remove an amino acid out from a chain, it means that the protein will not form. So it simply means that a mutation could result. It could result in the formation of an incomplete protein. So we would say that a mutation would take place and that it will not form the correct protein. It will not form the correct protein. And you can refer to that as a mutation, and that would obviously affect the protein synthesis. Let's look at the next question. What are asparagine and methionine collectively known as? And that's a key word here, collectively. We know that I've mentioned that asparagine is an amino acid. Both of them are amino acids. But the question is, what are they collectively known as? So if you said amino acid, well, you'll be correct because they are known as amino acids. But the question wants you to describe what they are collectively known as. Hope you guys are thinking about this. If we take two amino acids and put them together, so we get, I'm going to extend the page. So if we take one amino acid and join that to another amino acid, we got an amino acid number 1, an amino acid number 2. We call that, obviously they're held together by a peptide bond, and I know now you guys are guessing correct. So 1 plus another would be 2, so we call that a dipeptide molecule. Okay, meaning they have two amino acids held together by a peptide molecule. So the answer, this is quite a tricky question. Simply saying amino acids would not be, I would think would be not getting you the correct answer. Collectively known as would be dipeptide, meaning that they form a peptide bond to hold them together. We also get molecules that have several amino acids together, and we refer to that as a polypeptide molecule. So many amino acids held together forms a polypeptide molecule. Okay, so again we are going to skip through these questions. We've done a lot of questions on genetics. Okay, we're gonna go for a short break. But before we get to the short break, we still have some time for that break. So I'd like to do this question here before we get into the last segment. In this question, guys, here we're looking at an illustration that shows the separation of DNA fragments produced from blood samples at a scene of a crime. And there were 3 suspects here, guys. So we're going to play forensic investigator, and we're going to determine who the suspect is that committed the crime or that is linked to the crime. Guys, if you look at the diagram like this, the first thing that you need to remember that this is a genetic fingerprinting diagram, which simply means that it is a representation of strands or bands of DNA of various individuals that are used to compare relatedness or used to compare a match in terms of how closely they are linked to an individual. So we've got a very important key here. I'm just going to go up a bit. The key tells you that V is the victim, CS would be the sample that is found at the crime scene, S1, S2, S3 are the suspects that were possibly linked to the crime. So let's look at the questions. Okay, which suspect is most likely to be guilty? Now, how do I do this? Now, simply looking at it, if I stand back and simply look at it, it seems quite confusing. So guys, take a ruler. You take your ruler and you place it, and that's the band that we need to look at. So we've got the crime scene sample, and we've got suspects 1, 2, and 3. Use your ruler, guys, and run your ruler down and start looking at similar patterns and similar banding. Here we see that match with that. If we go down, we can see there's a band that is similar there. We can see that that's similar. So if we go through this entire, we can clearly see that although there are some similarities between S1 and S2 with the crime scene sample, there's an exact match of suspect 3 to the crime scene sample. Okay, so very quickly we're going to say which suspect was most likely to be guilty. It was the suspect 3. Okay, explain your answer, because we said that if we compare the banding patterns, guys, if we compare the banding patterns, we can clearly see that there is an exact match in terms of band for band that the samples match, and hence we can see that suspect 3 could be related to the sample matches suspect 3. Okay, so we've explained that it matches. Very interesting question. Would it be fair to convict the suspect mentioned in the previous suspect 3 on the basis of only the DNA evidence? And this is quite controversial, simply because in this case, if the suspect happened to have been there and for some reason maybe got injured while trying to assist the victim, he could have got hurt, cut, and the blood fell off there. It does not mean that he committed the crime. So he could have been there and maybe got injured while trying to assist the victim, and that's how possibly the blood sample is there. So we cannot conclusively use DNA evidence to link a person to a crime, but yes, we can use it to prove that he was at the scene of a crime. So it's very important. You would need to use other evidences to link him to committing the crime, actually. So it's not conclusive that we can use that. Before we go, the last question. Give 3 reasons for your answer, and we just gave you those reasons. Now I'm really excited about getting through this question here. Other than forensics, give 2 other uses of DNA profiling and fingerprinting. So the other uses would be determining relatedness. Parents often need to determine if kids are mixed up in the hospital, which doesn't often happen. You could use genetic profiling to determine whether the child belongs to which set of parents. It can be used for DNA screening as well, genetic screening of the possibilities of genetic disorders. Okay, and then finally, list 2 reasons why it would be an advantage to have a DNA profiling for a country. One is that we would avoid the theft of identity. We'd be able to easily keep record of individuals' genetic history. We can determine predisposition of diseases as well. There's also a flip side of it in terms of being a disadvantage in terms of invasion of privacy. But we're going to go for a short break for the last segment.
All right. right, Mindsetters, please stay tuned. We will be back straight after this. Welcome back, Mindsetters, from that very short break. I see some of you have questions, so I'll get straight into them. Kenzani is asking, when it is said that we must represent the information on a genetic cross, does it specify whether we should use monohybrid cross or the Punnett square?
So the question is, how do we represent a genetic cross? So remember I mentioned when we did a genetic cross, when we're representing a genetic cross, we start with the parents, P1, we say P1 genotype, we go down to the P1, we could have said P1 phenotype first, and physically how they look, phenotype, we go into genotype, we then go into the gametes, P1 gametes, and we say fertilization, which I missed out, and some of you asked whether it's important. Yes, it's important to write down fertilization. You then put down your fertilization using the Punnett square, and then you show your F1 generation, showing the genotype ratio, and your F1 phenotype ratio. So when you ask to show a genetic cross, you've got to show, look at the mark allocation. This could come for about 8 marks. Look at the mark allocation. If it is an 8-mark question, know that you've got to lay the question out simply that way. If you ask to show by working how you worked the probability of this, and if you see 4 marks, then it simply means that the question could read, show what is the possibility of the offspring being heterozygous. So you then need to use the Punnett square, so you're going to get marks for the Punnett square and for working out the gamete. So it might need you to go just with the middle, so the Punnett square, the F1 generation, and the parents and the fertilization. So yeah, a genetic cross would be, but a good indication would be looking at the mark allocation to determine the length of the layout of your answer. Was there another question, Looney?
Yes, I've got 2 more. Matsobane is asking, when doing the Punnett square, do we have to include fertilization, or does it have no mark?
No, yes, we have to, because just writing down the word fertilization is important, but the process is represented using the Punnett square. So it is important. I may have missed out fertilization. Really, you don't really need to have written the word down, but the representation of fertilization is by the Punnett square. So yes, you have to have shown fertilization via the Punnett square, not necessarily having written the word fertilization.
Okay.
And the other question?
And then Kabelo is just asking, can I write my life science essays in point form?
No, Kabelo. If you look at the last 3-mark allocation, it says synthesis of your essay. So your essay is, and that's what we want to see, your essay is basically how you present a scientific document. So you often respond, they could ask you, describe what stem cell therapy is, give 3 uses, describe the advantages, describe the disadvantages. So when you're doing your planning, make sure that you categorize those sub-criteria topics into different paragraphs. You expect it to write in paragraphs. This is not what is expected, but if I'm recommending this, if you for some reason feel that you've got 2 minutes left or 3 minutes left and you're not going to be able to write an entire page or page on your essay, then I will opt for very quickly putting down points, and that is simply to get down the information. But if you're writing an essay, if you've got time, you will lose marks if you've written an entire 2-page essay simply by bullet points, because you will lose the marks in terms of for synthesis, analysis, and presenting your information. So worst case scenario, 2 or 3 minutes left and you need to jot down points very quickly, then you do that. But you will lose out the marks for synthesis, introduction, conclusion, and stuff like that. Okay, so only if you are short of time. Okay, so we're going to go on to the next question, and that's basically a question from biotechnology, okay, or genetic engineering, adapted from September 2012. This is an Eastern Cape paper. It's nice to have a look at different questions from different examining boards. Okay, so here we're seeing the diagram below shows a modern technique to manufacture insulin. Study the diagram and answer the questions that follow. So here we're seeing a diagram of a bacteria. It's called bacteria E. coli. In it we're seeing this circular structure here, guys. And now we're seeing that circular structure being cut up by an enzyme. Then we see a human cell showing you the insulin gene in the DNA. There's the gene here that has been removed. An enzyme is then used to slot in that gene into the circular structure here, which is called the plasmid. So the plasmid is the vector that is used to transfer the gene. And here we have now, this is the recombinant plasmid or recombinant DNA, where we've recombined the gene from human insulin for human insulin production into the plasmid of a bacterium. Then we see further down, I'm just going to extend the page, go down a bit. We're seeing that the recombinant DNA is inserted back into E. coli, and then we have multiple copies of this, and eventually from these E. coli, recombinant E. coli, we have insulin being produced. So let's name the technique used in the manufacture of insulin. It's called recombinant DNA technology. Recombinant DNA and its technology, so it's a process, and hence it's called recombinant DNA technology. If we go down to the next question, identify part labeled A, and we looked at part labeled A. Part labeled A is a circular ring of DNA, and we refer to that as a plasmid, and we use the plasmid to our advantage to be able to transfer the gene into it. Okay, what type of cell division takes place at D? If we look at D, guys, D shows us from here, guys, if we look at D, from a single bacteria we have multiple copies here. For this to happen, you need cell division to take place, and that type of cell division is called mitosis. So mitosis produces several copies of identical cells with no change. Okay, and then we go down to mention two advantages of the above technique. To produce insulin, it is very costly. However, to use this technique, initially it's costly, but over long periods of time the cost of this becomes reduced, so it's much more cost efficient. It is also produces an exact copy of insulin, so the possibilities of rejection. In the past, insulin was used using bovine insulin, and that created allergies, it created negative reactions in the body, so individuals rejected them. However, it is exactly similar to how humans will produce insulin in their pancreas, and hence it reduces the possibilities of rejection. It is easily available, mass produced, it's cost effective. Okay, name another unicellular organism that can be used in the above process. The other unicellular organisms we use in this process are so this is a bacteria, could be viruses as well as yeast cells as well. So we could use yeast, we could use viruses as well. So viruses can also be used. If we go down to the last question in this section, give one reason for the use of unicellular organisms. Guys, if we want to manipulate an organism and change its DNA, the easiest way to do it is to use a single cell organism, because it's easier to change the gene of one cell than to change the genes of a multicellular organism. Also, it is easier because they reproduce faster, and hence you can get a recombinant bacteria reproducing many copies in a short span of time. We're getting down to the last question. I know we're pushing for time. I have to do this. It's on cell division, a very important topic. A substantial amount of questions normally come from this. The diagram below represents certain stages of meiotic division. Remember, there are two types of cell divisions, of which meiosis is important for you in grade 12 in a certain plant. Study the diagrams and answer the questions that follow. Here we see metaphase 1, clearly represented as metaphase 1, here anaphase 1. Okay, we're seeing that these two stages collectively give you an ovum, and this ovum is a diploid ovum. Okay, and here we're seeing a haploid sperm that comes together and give you a zygote. There's something not right in this diagram, and I hope you guys realize that this is a diploid cell that is produced after meiosis. Let's answer some questions that are based on this. Mention an observable characteristic feature of metaphase 1. Now guys, the word is observable. From this diagram, let's look at the diagram and see what is a characteristic feature of metaphase 1 from this. So for metaphase 1, we know that the homologous pairs of chromosomes, they meet or they align themselves along the equator. So the positioning of these chromosomes as pairs along the equator is a characteristic of metaphase. So they meet as pairs, and that's very important. Remember that it's not just on a single line. Here they're meeting maternal paternal as a pair along the equator. So fundamentally important. So they meet as pairs. So it's homologous pairs along the equator is a characteristic feature of metaphase 1. Name the process that takes place in prophase 1 that leads to variation in the offspring. Guys, remember that prophase 1 is this first stage after interphase in meiosis 1. The process that results in variation is called crossing over. And that crossing over simply means, I'll show you a very quick representation. It is that process there. And an important point of crossing over is the chiasmata. So we've done two concepts. The process that results in variation is called crossing over, and the point at which it takes place is called the chiasmata. Also remember that variation can also take place during random assortment, during anaphase. When they move apart, there's random assortment of the way the chromosomes move apart. Also, there is independent random arrangement along the equator in metaphase, and independent assortment, the way they move apart. So it's maternal paternal, paternal maternal. So that combination varies. We're going to do a few more before we break. What error occurred in anaphase 1? Looking at this diagram, guys, here we're seeing we've got a space there. It simply means that the homologous pairs have pulled to one side, which means that there is non-disjunction. The word is non-disjunction, or non-separation of chromosomes at the equator. So that's a very interesting question. What will be the chromosome number in AB? Okay, so we're going to very quickly look at that. But you guys can have a look at these. You can download these notes. We're actually coming to the end of the session. It's gone very fast. All the best for your prelims. I'm going to hand you over to Looney.
All right. Thank you so much, Mindsetters, for tuning in. Remember, the notes are on our Facebook page, so check them out there, facebook.com/learnextra. Everything of the best for your prelim exams. Write legibly. Remember to focus. Concentrate on the questions. Do not panic. Nervousness will take over you and you will just be in a fluster. Don't panic. Read every question carefully, and everything of the best, guys. Till next time, goodbye from us.